Good morning, grade 12. I have sent one small video regarding the restriction endonuclease. You just go through how the pictures will be and you can able to follow the slide what I'm going to show now. So we know what is biotechnology. Quickly we'll reveal what we learned. Bio means living, technology means we are using techniques to get some useful products. And what is EFB, European Federation Biotechnology, what they have given regarding the biotechnology definition. And oldest form of biotechnology is making breads and making curd, other name for curd, or when we make curd in an industrial huge level, that we can call it as yogurt. So making breads and curds with the help of microorganisms. For example, if it is a bread, it is a yeast, single cellular fungi, Saccharomyces cerevisiae. If it is a curd or yogurt, lactobacillus bacteria. So here bacteria, here fungus. So this is example for oldest form of biotechnology. Apart from that, fermentation, making of wines, alcohol. So they didn't use much techniques like molecular level. They didn't use any technique. Simply with a starter culture, like curd means if they are introduce a small amount of curd into the milk, it will get into the it will change it into curd again. So milk changed into curd, that we can call it as a older biotechnology method. They didn't use so many different techniques. Without knowing even, they did all those things. So that is also nothing but biotechnology only. And what are the two importance we learned? Genetic engineering and chemical engineering. Genetic engineering means here we'll change the DNA and RNA in some of the sequence and will make so many new things. A recombinant DNA technology comes under genetic engineering only. And chemical engineering means here only recombinant DNA and we can able to get our desired things. And in chemical engineering, gene cloning is involved here, and there what and all are useful products like vaccine, enzymes, beverage, drugs, not only for human being, it is also for cattle. Like some of the hormones they'll use for even for cattle to induce milk. For example, some of the hormones, somatotropin, okay, bovine somatotropin is a hormone which will increase milk in the cows. Like that, so many hormones they'll use for even cattle also. So we are getting benefit through biotechnology. Not only we are applying for cattle also to get benefit for us. So that's all comes under chemical engineering. And we learned all those technical terms: recombinant DNA, gene cloning, gene transfer, and what do you mean by origin of replication. Okay, this and all technical term. The same I will explain in a detail while explaining the concept. So the technical term is clear. And what do you mean by vector? We saw one such example for that as a plasmid vector in a Escherichia coli. It is naturally present extra chromosomal circular DNA. And all those recombinant DNA, what technology and all we learned. And what are the three steps to form GMO, genetically modified organisms? First we need to identify and we need to introduce into the host, and host will have a now recombinant or genetically changed organisms, and we should check whether they are GMO and we need to maintain that. These are the some steps to get GMO organism, genetically modified organisms. Normally GMO we can try for plants, animals and microorganisms, not for human beings. So when we try for plants, that we can call it as transgenic plants, because we changed a gene, some of the genes. So we can call it as transgenic plants. When we change some of the genes in animals, that we can call it as transgenic animals. And when we change some of the genes in microorganisms, that we can call it as genetically modified microorganisms. So we won't try for human beings. Normally human beings, gene therapy is applicable. Will identify a disease gene and will change the disease gene in the human being. That is only they are doing in biotechnological. So they didn't change the totally, they till now they didn't change totally the genes of the human being. They are trying genetic engineering for human being only to cure disease. Gene therapy only they are using. And for plants, animals, microorganisms, totally they have changed some of the genes and they formed a genetically modified organism, GMO. So you want to remember transgenic plants, transgenic animal, GMO, genetically modified microorganisms. So if genetically modified microorganisms, it is all for our purpose. So we can harbor or we can harvest our useful products like vaccine, hormones, antibiotics, like that. Whatever we need, we can harbor from the, or we can harvest from the genetically modified microorganisms. So till now we saw, I think you are clear with that. And what is the step behind that? That is clear. And this slide onwards I'll explain afterwards, after thorough with the tools of genetic engineering. I think you may able to follow all those things. Now this is only we are going to see now in today's class onwards. So recombinant DNA technology is accomplished with tools. So here tools means don't think only the mechanical tools. What and all is very important for genetic engineering. All those things together we can call it as a tools only. Now first one is restriction enzymes. Okay, first one is restriction enzyme. So here I have sent a previous video regarding endonuclease enzyme. So restriction endonuclease, restriction endonuclease, exonuclease, and single endonuclease alone. So with that I told exo and endonuclease, it is not much applicable in biotechnology. Only restriction endonuclease enzyme is only applicable in biotechnology because it has its own specific function. So how it means that I'll explain later. So in biotechnology we are very particular with the restriction endonuclease that we are obtained from mostly from bacteria, that two eubacteria and archaebacteria. Actually these restriction endonuclease are the chemical weapons or defense mechanism which is present in the bacteria to prevent the entry of virus or other foreign materials to not disturb the organisms. So when it enters also, these enzymes will cleave the DNA. So once the DNA was cleaved, automatically they can't able to multiply inside the bacteria. So the bacteria will escape from the virus and other foreign substance. So these enzymes are defense mechanisms or chemicals which is present in bacteria, but our purpose, we are isolating these kind of enzymes from bacteria, and we are using in laboratory for our purpose. So how we are taking these kind of enzymes from the bacteria means, for that I have given some of the enzymes that I'll repeat once again. First we'll finish this one. So now restriction endonuclease is clear, and exonuclease, when we need only the blunted, we can go with the exonuclease. So endonuclease between DNA sequence, exo only the extreme DNA, and that too a restriction endonuclease is very particular between in some areas. Okay, they will cut both the strands of DNA. Only endonuclease means only one strand it will cut. Okay, is it clear, children? Exonuclease means end, not both the end, any one end it will cut. Endonuclease means any a middle part of the two strands, but restriction endonuclease alone, it will cut both the parts of the DNA in middle regions. Okay, next polymerase enzyme. So here the name itself we can guess. Polymerase means it will help to increase the number of copies of DNA inside the host. So that polymerase enzyme, that is also we are taking from the one bacteria only. Okay, we'll call it as a eubacteria or archaebacteria. In that one interesting polymerase, call it as TAC, TAQ, TAC polymerase. Okay, TAC polymerase enzyme. What is special about TAC polymerase enzyme? So I told particularly archaebacteria. Okay, archaebacteria. So normally we used to take TAC polymerase in archaebacteria. So we know archaebacteria can able to withstand in a high temperature. So if it is a high temperature also, it can able to withstand means. For example, say example for one archibacteria, what we learned in 11th standard, that is Thermophilus aquaticus. That is a archibacteria that can able to withstand a high temperature, even 100 degrees Celsius or more than that also. So we are doing this recombinant DNA and gene cloning in a laboratory. So in one condition alone, they'll make this equipment at a very high temperature like 90 degrees Celsius to get more copies of gene cloning. So for that they should use only Taq polymerase, because Taq polymerase can able to withstand the high temperature, and it will keep on give instructions to the DNA to make more copies. Okay, it is an enzyme. What is the enzyme? Enzymes are biocatalysts, so they'll help to speed up the rate of the reactions. So this Taq polymerase, which we are taking from the archibacteria like Thermophilus aquaticus, it can able to withstand high temperature like 90 to 180 degrees Celsius. This PCR, polymerase chain reaction, needs high temperature to get or to make more copies of DNA at a time. So in that condition we always prefer Taq polymerase. So this Taq polymerase we are extracting from a bacterium, call it as Thermophilus aquaticus. And third one, DNA ligase, it is help to join two DNA strand, and vectors only are plasmids. Okay, we want to see in detail. And what is host might be microorganisms or other host, okay, plant or animal. So these are the 5 tools what we are going to learn in a detail. So everything we need one video. For restriction enzymes, to explain that even I need one video lesson. I can able to complete within 10 or 15 minutes only. I need 10 to 15 minutes to complete restriction enzymes. So like that we want to see 5 also in detail, and afterwards you will be thorough with what do you mean by biotechnology and how they are using in techniques. Now is it clear, children? So restriction enzymes, endo or exo. Polymerase enzyme, that is also we are extracting from bacterium. Polymerase means it will help to increase many copies. So what is the instrument used for that? That is PCR, polymerase chain reaction. So nowadays to check whether coronavirus is positive or negative, they are using RT-PCR. It is reverse transcription polymerase chain reaction. Why? Here we are talking about DNA only, but that is virus. Virus means it will change into DNA, and then only we can able to go for this technique. So that is reverse transcription PCR. Okay, I'll explain. PCR is in our syllabus. I'll explain the concept. And ligase is an enzyme used to join the DNAs, and vectors, it is a vehicle to carry the recombinant DNA, and host where the multiplication will continue. Is that clear, children? So next we'll move on to the first enzyme, restriction enzyme. Is it clear, children? Now listen this video and go to your previous video what I have sent already. That also you listen and wait for some minutes. I'll send next video for this restriction enzyme in a detail. Thank you.